have a model of a muscle fiber, a single muscle fiber or muscle cell, and some of it is sectioned and cut away so that we can see inside. And you can see each of the individual myofibrils, and that would be these from the top-down view. And you can see them also in the longitudinal view. And you can see here's a muscle myofibril, and here it's cut away to reveal the myofilaments, which are the actin and myosin. And of course, as you remember, the myofibrils are arranged into sarcomeres, which are the unit of contraction of the muscle cell. So you have your dark bands, which are where your myosin filaments are, and you have your light bands, your I bands, which are where your actin filaments are. And the dark bands are also sometimes known as the A bands, and the light bands are sometimes known as the I bands. In the middle of the dark band, you have the M line, and in the middle of the light band, you have the Z line. And remember, the sarcomere is delineated by two Z lines. And when this thing contracts, the Z lines will move towards the M lines as the actin filaments slide over the myosin filaments. We also have, if you look on the outside of each myofibril, you see this lacy beige stuff. This is the sarcoplasmic reticulum in which the calcium is stored. Notice these thickenings of the sarcoplasmic reticulum right here, where you've got these bulges that encircle the entire myofibril. These are the terminal cisternae. So this is one terminal cisterna, and here's one on the other side of this blue thing, which you will probably recognize as the transverse or T-tubule. Now, as you recall, the sarcolemma, that is the membrane of this thing, that is delineated by this pink part. You can follow it up along the sides here as well. So this little thin line here, cut in section, is the sarcolemma. And this whole thing, remember, it is the membrane of the cell. And notice how it has these indentions inside that lead into the T-tubules. And the T-tubules, the transverse tubules, are sort of indentions of the sarcolemma into the cell to carry the action potential into the cell. As you recall, a muscle membrane is an excitable membrane, and it can be stimulated by a motor neuron. That would be this thing right here. This is our yellow motor neuron right here, and this cell here surrounding it is a Schwann cell. You can see the insulation and how it's wrapped around the yellow part, which is the motor neuron, and it is stimulating this neuron or this muscle cell at the neuromuscular junction. And so we see here where the yellow is in close opposition to the sarcolemma, we have the neuromuscular junction. And that's all of this here. And notice how the sarcolemma takes on this kind of squiggly shape here. And notice also that we have these little vesicles, these little white blobs in here, which are synaptic vesicles. And these are full of acetylcholine. So as you recall, the action potential comes down. The motor neuron causes the release of the acetylcholine by fusion of the vesicles with the membrane. The acetylcholine then diffuses across and binds to receptors on the sarcolemma, which then opens sodium channels, and you know what happens from there. That generates the action potential, which is propagated down the sarcolemma into the T-tubules, where they cause the release of calcium. The action potential causes the release of calcium from the sarcoplasmic reticulum, most specifically in these terminal cisternae. So you'll notice the arrangement of the terminal cisternae around the T-tubules. So each one of these T-tubules surrounds the myofibrils as it comes in. And you can see that the sarcoplasmic reticulum is arranged around it. So you've got a terminal cisterna on either side of these T-tubules. And this arrangement is called a triad, where you have two terminal cisternae sandwiching a T-tubule. And as you'll notice, they're typically right over what would be the zone of overlap. You can't really see it here in this model very well, but you know within the dark bands, some of the actin is going to be interdigitated in here. It's not really shown on this model, but the zone of overlap is right about through here. And depending on the state of contraction, the zone of overlap will get larger, and then in relaxation, you'll have less overlap. Now, the other parts of this that we need to know are these red things everywhere. These little squiggly inside red bean-shaped things are mitochondria. And they're necessary both for the neuron and for the muscle itself, the muscle cell, because these processes of stimulation and contraction require a lot of energy. Now, as we remember from skeletal muscle, they have lots of nuclei. Here is a nucleus of a skeletal muscle, and they're located peripherally around the edge of this thing. The other thing that we would need to know is the Endomysium, which surrounds each muscle cell, each muscle fiber. And that's this white stuff around here. You've got the Endomysium, which is that connective tissue that surrounds each muscle fiber. So that is the Endomysium. And then we can also see that if you look in section here, you can see little bits of T-tubulae. This isn't a transverse section. It will be coming out of the plane towards us. And since this has been sectioned, like this one, we can see inside of it. And we can also see the Sarcoplasmic Reticulum and the terminal cisternae on either side as well. So this is an interesting arrangement, and it allows for the conduction of the action potential as the muscle is stimulated by, or the muscle cell is stimulated by this motor neuron here. The T-tubules bring in that action potential that was initiated within the sarcolemma, bring it into the interior of the cell where it can reach the sarcoplasmic reticulum and cause them, these terminal cisternae, to release their calcium ions, and then you know the rest. So that concludes our tour of the muscle cell model.